Hey, so welcome back. It's our second e-pitch. Today it's by Kunal Kadiya. Kunal is a Ph.D. candidate at the University of Saskatchewan in Saskatoon, Saskatchewan. His research areas include developing food-grade nanoemulsions, exploring the use of protein and biopolymers in making bilayer emulsion gels, and studying their rheology and digestion behavior. Kunal received his master's in dairy science from the National Dairy Institute in India, worked on the technology to develop a probiotic cheese as part of his master's research. The title of Kunal's presentation for today is Effect of Interfacial Composition on the Rheological and Digestion Behavior of Emulsions. So welcome, Kunal.
Thanks, Doc, for the introductions. Hi, everyone. So today I'm going to deliver my ePoster pitch on effect of interfacial composition on the rheological and digestion behavior of emulsions. So we know that we consume many food emulsion cells in our day-to-day life, but typically if I'm talk about the emulsion gels like a mayonnaise, which has around 80% fat, the close packed structure created by the repulsive droplet, it's giving the food gel kind of structure here. In my research, the goal is to create the mayonnaise-like structure with low fat. Particularly in mayonnaise with 80% oil, the effective volume fraction is very similar to the actual oil volume fractions. And here is the equation how we can calculate the effective volume fractions. But my objective is to reduce the droplet size here and then increase the interfacial thickness so that I can get the similar kind of structure at lower oil concentration by increasing the effective volume fractions. My hypothesis is that by reducing the droplet size from coarse emulsion to the nanoemulsion, and then by putting the second layer of biopolymer onto the nanoscale droplet, we can increase the interfacial thickness, and this will allow the droplets to close pack, and those close pack structure will not allow the droplet to move, and the droplet would be, create a gel kind of structure from the liquid emulsions. To illustrate this one in a simple way, I have a really good example here. So we can see here the herd of sheep where without the wool on their body, actually, they can move really easily. But think on the other sides, when they have really thick wool on their body, they cannot move easily around. So that's the importance of the wool thickness there. Similarly, in my research, actually, the interfacial thickness can play a similar role, and the close pack structure created by a much higher contribution of the interfacial thickness can lead to the gel kind of structures in the emulsions. Another advantage of creating a gel-like structure would be, we hypothesize that the digestion behavior, the digestion would reduce in that case when we have a thick interfacial layer, because our hypothesis is that the lipase actions on the triglyceride molecule will restrict because of the higher interfacial thickness, which will delay the lipase action, and there is possibility to reduce the digestion of the lipid in that case also. So with that hypothesis, how did we reduce the droplet size to the nanoscale? So we used a very surface active food grade emulsifier, Citrem, with 40% oil, and we used the high pressure homogenizer to reduce the droplet size to nanoscale around 200 nanometers. Our second approach was to reduce or to increase the interfacial thickness. To increase the interfacial thickness, we used the positively charged chitosan polymer to deposit onto the negatively charged Citrem-stabilized droplet. And by increasing the interfacial thickness, we just want to achieve the close pack structure. And for this particular research, we used a different concentration of chitosan and with different positive charge of the chitosan, that is the degree of deacetylation. But for the time's sake, I will just presenting the results using the DD93 chitosans. Come to the results. So gel strength. So we compared the gel strength by plotting the storage modulus as a function of chitosan concentration. So higher the storage modulus, then higher the gel strength of the emulsions. We can see with the monolayer Citrem-stabilized emulsion, the emulsion was kind of liquid. But with the increase in chitosan concentration, we can see that we were able to create the gel kind of structure with higher storage modulus. And then eventually at 0.25% chitosan, we can see the storage modulus still reduced compared to the intermediate concentration of chitosan. This could be understood by the confocal microstructure of these emulsions. And at the bottom, we have the droplet charge and the droplet size. So we can see that at 0% chitosan, when only Citrem-stabilized droplets were there, droplets were very nicely distributed with the negative charge and with the lower droplet size. But as we added the chitosan, the droplets were flocculated, probably because the polymer concentration was not enough. That leads to the bridging flocculation among the droplets, and that's created the aggregated structure or attractive emulsion gel at intermediate concentration. But as we increase the chitosan concentration to the higher concentration at 0.25%, we can see the droplets was positively charged, repulsively charged, and again we can see the decrease in droplet size. So this close pack structure created by the positively charged and the higher interfacial thickness can lead to the close pack structure, and the droplet cannot move, and it's actually increased the storage modulus around 10 times than the Citrem-stabilized droplets. Another objective was to evaluate the digestion behavior, and we analyzed the digestion behavior of emulsion by following the InfoJust2 protocols and by PSTAT protocol. Actually, on the right side, we can see that the fatty acid profiles of the emulsions at different stages of digestions. We can see that the digestion of the emulsions reduce as we increase the chitosan concentration, and when the droplets are completely covered by the chitosan, the digestibility was reduced from 25% to the 17%, and that's because of the restrictions on the lipase actions on the triglyceride molecules. To summarize these things in terms of the gel strength and lipid digestibility, by this research, actually, we got three different kind of structures. Liquid nanoemulsions, when the droplets are only stabilized by the Citrem only, that's the monolayer emulsions. But as we added the chitosan, we can see that the storage modulus increased, and the kind of gel we got, it was because of the bridging flocculation. But at higher concentration, the droplets were completely covered by the chitosan, and the increase in repulsive nature between the droplets and increase in the interfacial thickness leads to the increase in the gel strength and the kind of gel structures. The similar effect we observed on the lipid digestibility, as we have like the droplets were more and more covered by the chitosan, the digestibility was reduced, and it was reduced around like from 25 to the 17%. So the takeaway message from my research is that by creating this kind of structure, it can be used into the low-fat products with the controlled digestibility. Thank you for your attention, and I'm happy to answer your questions.
Thank you, Kunal. That was a great presentation. Really enjoyed it. Some really good science there. So, judges, do you have some questions for Kunal and his paper?
Yeah, Kunal, I think it was very interesting, because when I look into mayonnaise, it's definitely tasty, but definitely also high fat. So often I wonder if low fat ones can also taste as good as the high fat ones, right? It's interesting, so when we talk about mayonnaise, normally people only use liquid oils, canola, sunflower, soybean, and in many parts of the world, those are very expensive oils, while palmolene is very cheap. But palmolene still has a bit of solids that does not sit well in an emulsion like mayonnaise. So do you think this kind of double layering, we can prevent a bit of, what do you call it? I don't know if it suppress crystallization a bit, so it does not connect and separate. Have you tried?
So we use the canola oil for this study. So we didn't have any experience with the other oil, but it would be really great to, look into that part as well. And probably it is possible to, creating this kind of interfacial thickness to, what you are targeting, that can be also achieved. There are many other aspects of, by coating the droplet. This is kind of the encapsulation process. You are just giving the high interfacial thickness to the droplets, and our main objective is to just look onto the gel strength and then the digestibility. But that can also be checked in the future, actually, with the different.
Well, I think from the industry point of view, I mean, for customers would be quite interested if you can suppress so it does not crystallize out of the droplet, right? Another question, what's the main role of the Citrem, do you think, here? Because you added 3%, that's a lot of emulsifiers. It's good for our business, but people don't buy that much amount.
Yeah, so for today's presentation, I have the time limit. Otherwise, my whole research, and even I published the paper on this, actually. So we used 3%, but we removed the excess Citrem from the continuous phase. So the role of the Citrem is that it's first we wanted to make the food grade emulsion actually. So that's why we use the Citrem. And Citrem is a really surface active molecule to reduce the droplet size to nanoscale. But the science says that you need really high amount of emulsifier when you want to reduce the droplet size to the nanoscales. And that's the problem when you want to put the second layer onto the droplets. Excess emulsifier actually into the continuous phase can react with the polymer first, so your polymer cannot go to the interface. So what we did actually, we removed the excess, and then we were able to deposit the second layer onto the droplets. So although it's a 3%, but I have the data, and the data says that I have removed around 1.2% of the Citrem from the continuous phase. So probably in the secondary emulsion, I have around 1.5 to 1.6% Citrem onto the droplet actually.
And I think just one couple of comments that, if you can also try datums. They're also very surface active. In fact, for mayonnaise, we promote a lot of datum instead of Citrem because when you talk about Citrems, you need to identify what kind of Citrem was it, what kind of monoglyceride was there, or what's the acid value. So Citrems can have such a wide range. So maybe in the paper you did, but in this short scope you didn't. But
definitely, so I can answer that question actually. We use the Citrem with the HLB value of 9 to 10. So it was kind of intermediate range, which has really surface activity actually compared to the lower HLB value. Actually.
Focus on the acid value. HLB is pretty empirical. Acid value will tell you how much amount of actual citric acid esters are there. But that's a different discussion. But very good. Very nice. Thank you.
Thank you so much, Gus.
Kunal, thank you. I really enjoyed that as well. You made excellent use of your schematics and visuals. The sheep diagram in particular was very telling to demonstrate the bilayer effect. And your summary slide where you overlaid things in the schematic to really integrate your findings, I thought was especially well done. So I enjoyed it overall. I was wondering if you could comment a little bit more about your digestibility differences and why you might be targeting decreased digestibility in your as an end goal.
Yeah, so we want to just reduce the digestibility. The goal is that actually. So suppose if you are taking the food, but when you take the high calorie food, definitely the digestibility, in terms of I'm talking about the calorie you are taking. So here the thing is that if you have the control over that release actually during the digestion, definitely the energy intake. So the human beings are such that they are working and they are consuming the energy from the food actually. So the thing is that we just want to control that process actually. So the second layer, the chitosan, or maybe we can put some other interfacial composition. So this is really generalized statement. When you have really thick interfacial layer, then it is possible to reduce the digestibility or control the digestibility. This concept is also used in many other applications for the control release of the bioactive molecule from to the droplets. So that's what our objective actually. So to get the gel strength at lower oil concentration and then to reduce the digestibility. So that would be kind of, the functional food in that case to reduce the calorie content.
Yeah, some people might challenge to say, well, do you really want to prevent too much digestibility? Where will that go? I understand what you're saying about from a caloric perspective, but what are our systems made to handle a whole bunch of undigested lipids in the lower intestine, for example?
That's a really good question actually, and we didn't look on that actually. So this was a kind of in vitro digestion, but that would be really great to start try this kind of emulsion in an animal in vivo study as well. So probably that would give you a better idea like how these emulsions are behaving actually.
Yeah, I was particularly interested when you said to control the digestibility. I mean, maybe not to stop digestibility per se, but in order to tailor it so that you might have some sort of controlled release, especially as you said, with respect to the bioactives. So thank you.
Thank you so much, Emma.
Okay, Kunal, also from my side, many thanks for your pitch. I enjoyed also the visuals, like Amanda already said.
Thank you.
And I also love the nanostructure approach to create these microstructures. You showed some confocal microscope pictures, but of course you do not really see like a bilayer in those pictures. Did you ever try to also visualize with other
Yes.
techniques,
So we
the
have
nanostructure?
Yes. So we have the SCM images actually, and we can really see the second layer onto the chitosan second layer onto that. And to, in the confocal structure, actually, we also added the another dye, fast green, which can really react with the chitosan, and we can see those structure. But the thing is that the kind of confocal we have in our university, it's not that really great to focus onto the interface specifically. So we are trying to get those data by SCM actually. Apart from that, we are also trying to look onto the structure using the USASC actually. So we already did that study. We have the data, we are analyzing those things. So probably in the future, we'll come out with the paper where we'll come out with the thickness of interface and then how the chitosan is actually contributing into that.
Okay, and that's possible with USACs to get some information from that, or?
Yes, we are targeting that thing, and we are looking forward to that actually.
Okay, looking forward to that those results. You're using Citrem.
Yes.
I was just wondering, because there is a huge clean label trend in industry, are there clean label alternatives for this type of emulsifier to produce nanoemulsions?
Yeah. So Citrem is actually food grade synthetic. It's a synthetic emulsifier, but it's a food grade. And there was a project actually Nestle did with some university actually, and Citrem-stabilized infant formula actually. And they did the study for this one, and they said that it is really easy digestible. It doesn't have any adverse effect. So, and those data are already published on that. And based on that, we also tried to use the Citrem actually.
Okay, but you don't know that there is an alternative for
alternative is there actually. So basically, actually, we want to make the two layer. So the first layer should be negatively charged droplet. And for that, we need the emulsifier, which gives the real nanodroplet actually. So people use the sodium todecyl sulfate for a negatively charged droplet, but that's not a food grade. So the alternative we found is the Citrem actually.
And the chitosan, does it have any nutritional property or advantage?
Chitosan is like nutritional in terms of like it has the it can help to reduce the reduction in the cholesterol I heard about, but not in detail actually, and we didn't study about those things. But the focus for this research is to use the chitosan because there is only one biopolymer available which is having the positive charge, and to coat onto the Citrem we need the positively charged biopolymer. So that's how actually we use the chitosan without digging into the details of how chitosan would be helpful in the health benefits actually.
Okay, thank you.
Okay, thanks, Kunal. Great discussion, good questions. And we'll move on to our next speaker.